Hello, in this video we will understand about the complement system and the complement cascade. So what is the complement system? Complement system is the innate immune system which that is used for the opsonized pathogen to neutralize and the burst of the cell. So what is this? Let's begin to understand. It is a part of immune system, remember. And this is the innate immune system will never change ever. And that enhance the complements that ability, the complements protein, that ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism also. So basically it is promote the inflammation to produce the cytokines and attack the pathogens cell membrane and also recruit the neutrophil, basophil and other type of cells also due to the inflammation progression. Remember, let's begin to understand from the liver because it releases from the liver this complements protein, and this complement protein is 50 proteins and proteins fragments make up the complement system including serum and cell membrane proteins. So remember, the cell membrane also contain a complement protein. Remember sometime. But we will concern with the serum. The serum contain a complement protein also. So what is the serum and what is the blood, RBC, WBC? This is the serum. Serum contain a fluid as well as complement proteins and blood. Blood during the centrifuge, the blood will be moved down while the serum will be moved up due to the low density of the serum. This is the cell and this receptor protein. This receptor protein is the complement protein. Let's begin to understand the complement pathway. The complement pathway is contain classical pathway. We can divide it. Lectin pathway, alternative pathway. The classical pathway and lectin pathway and alternative pathway we will discuss differently separately, but we should forget about the lectin pathway. We will understand about the classical pathway and alternative pathway. This is the blood circulatory system contain microbes. Whenever the microbe will be enter into the blood circulatory system, the classical pathway or alternative pathway will be enhanced. In this way, the microbes contain epitope. Epitope is the antigen. So in this way, the whole microbes is the antigen. Antigen, in this way, the adaptive immune response will create antibodies against this antigen like IgG. After these antibodies, the Fc portion will be bound with the C1 complex. This is the complement system, the complement protein. C1 contain a C4. This basically is the C1 contain a lot of proteins, but the C4 is the protein will lead to cleavage of the C4 protein to produce two fragments like C4b and C4b. This C4b will attach with the 2A. In this way, the C4b will be attached with the 2A. And this 2A after the fragmentation due to the A convertase enzyme, the CA convertase enzyme. In this way, the half protein fragment will be attached with the antigen while the half will be attached with the complement system, C1 complex. In this way here you can see the C4b and 2A will be bound while the C4b and 2A will be bound with the antigen. Let's here is the C3 convertase will lead to cleavage the C3 protein into the C3b and A and 3A, sorry. So the C5 convertase enzyme also further going on. Let's begin to understand. Here is you can see this is the again I am drawing, and in this way the C4b and 2A already bind will lead to also attach with the 3b due to the fragmentation of the 3A, and in this way the will be formed here, and after this a C5 convertase enzyme used to fragment of the C5b and 5A. Anyhow, here I am drawing again the microbes. the IgG will be bind with the C1 complex. In this way, the C4b and 2a will be formed already, and in this way, the C3 convertase enzyme will be used. C3 convertase enzyme used to produce the fragmentation of the C3 into the C3b and 3a. This will be attached with the 3b, basically C3b will be attached with the 2a site with the antigen. So remember, in this way, the whole microbe will be attached with the membrane. Let's begin to understand the alternative pathway. Alternative pathway, again the C3 fragmentation to form a C3b and 3a, and in this way the attachment again part we will understand shortly, and C3b and bb will be formed. In this way the current C3 convertase enzyme used to produce the C3 fragmentation. This C3 complement protein fragmentation to form a C3b and 3a. This C3b and 3a will be again attached the C3b, C3b and bb and 3b. In this way, here you can see this is the going on, and in this way the C5b and 5a will be formed. Anyhow, the C5 convertase enzyme. The C5 convertase enzyme used to convert the C5 into the C5b and C5a. And after this, the cluster of the fragmentation will be formed to a complex formation C4b, 5b, 2a and 3b complex will convert into the C3b, 5b and bb and 3b. In this way, the C6, 7, 8 complement protein will be attached again and again one by one, will lead to 5b formation, and the C1, 8 and 6 formation, the poly C9 formation to the fragmentation 7, 8 and 6 to the 5b form the membrane attack complex. So in this way, remember, this is the cascade of event of the complement system. Remember, in this way, let's begin to understand again the alternative pathway with the explain differently. This is a C3 hydrolysis will be occur. In this way, C3b and 3a will be formed. The C5 will be cleavage due to the C3b attachment to cleavage the C5a and 5b. And in this way, the C5a will be attached as a complex, and in this way the C5b, 6, 7, 8, 9 cleavage formation will lead to attach in the membrane complex formation to cell swell and burst, and in this way the cell will be burst and together form the cylindrical membrane attack complex formation. Let's begin to understand through plasma membrane. Here the C3 will be contain hydrolyzation to produce C3 and BB attachment with the B fragmentation to BA as well as BB. So the BB fragmentation will attach with the C3, and in this way the C3 will be hydrolyzed further to produce C3A and C3B. C3B will be attached in this way. The further going on the step 4 from the 3. In this way, the step 3, the C3b will be in the presence of B convertase to BA formation and BB formation and C3b and phosphate. And in this way, the C3 and C3A, basically the B and BA will lead to produce, and in this way, the C3 and the C3A. C3 and C3A will form step 5, the B and small b, and C3b to C3b and phosphate group attach, and in this way the C5 convertase enzyme to produce C5A and C5b. And in this way, this is the cascade of phosphorylation, and the cascade of event of the complement system will lead to create a cylindrical membrane attack complex will form, and in this way the cell will be swell and burst, and after the bursting of the cell also the inflammation will recruit the macrophage for endocytosis or engulfing of this opsonized pathogen after the death of this pathogen or infected cell. Thanks for watching. Please make sure to subscribe, like and share. Bye.